Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Matt Lavoie, who is the Fixel Endowed Chair in Neurology at the University of Florida College of Medicine. Matt is also a 2022 Toffler Scholar. Today's discussion is focused on cellular mutations that may cause Parkinson's disease and the possible related pathologies of Lewy body dementia. So let's dive in. Matt, welcome.
Hi, thank you for having me.
Yeah, I'm excited for this and to just hear some updates of where you're at in your research and just talk about what you're doing. So let's start with just kind of laying the foundation for what got you into research and why the focus on this disease.
So I guess the focus on research began when I was pretty young. My father is a medicinal chemist, or actually was. He just retired a year ago from Rutgers College of Pharmacy. And at a very young age, I would go into his lab as a little kid on weekends when he was doing work, or watching him write grants and papers, prepare talks. And so I was fascinated with the field of science and academic medicine and research for a very long time. I don't think chemistry really suited me all that great. I dabble in it a bit from time to time, but as time went on, I found a fascination and a love for biology. I had a really fascinating biology teacher in high school that really inspired me that broadly biology would be where I would wind up in, and wound up in a graduate program that had very strong Parkinson's component, and really enjoyed my training at the University of Pittsburgh with Terry Hastings. And as my second mentor called it, my first love, I trained in Alzheimer's disease as a postdoc, and then I quickly went right back into Parkinson's disease. And Dennis Selko said I went back to my first love, and with all of the new genes and information we were gaining about the genetics that underlie Parkinson's disease, I got really excited about the opportunity for new discovery and to leverage all the skills I had learned in the Selko lab at Harvard Medical School, and to bring that experience into Parkinson's disease, things that we didn't have when I began.
So tell me a little bit about the Fixel Endowed Chair. Like, what does that, for some of the listeners, they may not know the significance of what that means. Can you talk about what that is and why that's really an honor?
So, well, it's a fantastic honor to hold one of the Fixel chairs. The Fixel family had gave a large gift to the University of Florida in early 2019 with the goal of recruiting 4 new faculty to the University of Florida to dramatically expand the portfolio of research that we were doing here in Parkinson's disease. And so when I got the phone call from Michael Oken in 2018 to see if I was interested in relocating my research program and joining the community here, it was a real honor to be part of something that has grown astronomically since my arrival. We are continuing to expand, bring in new faculty. I was here for the beam signing for opening a new clinical building on Williston Road, the Fixel Institute. And I see the Fixels when they come to Gainesville from time to time. And so it's been extraordinary to be a part of that really family of patients, those invested in improving patient care, the clinicians here, and really the whole enterprise around of movement disorders, and really expanding now to neurologic disease. And so it was exciting at the beginning, and it's continuing to expand. We're not done. And so it's a constant challenge, but it's very, very exciting to be a part of this moment of extraordinary growth here at the University of Florida.
Yeah, that's exciting. I know I've talked to other researchers that are a part of a lab or just getting started with their lab. Some are very small, some are large. Can you give me an idea of what your lab, what is it comprised of? How many people? And really the focus. Is it just Parkinson's or is it broader than that?
Sure. So my lab we have 3 junior faculty in the lab, as well as a graduate student, 2 technicians, and I have a senior lab manager with 20 years of experience in the study of neurodegeneration. And then we have a large cohort of undergraduates. So the University of Florida has outstanding undergraduate students, many of which are looking to gain research experience, to be involved in a lab, to figure out whether their future is in medicine. Do they want to go to medical school? Do they want to go to graduate school? And so back when I was at Brigham and Women's Hospital in Boston or here, the lab has always been a home for junior folks to come through, get a little bench experience, get some training on what we would call translational neuroscience, a focus on disease, and decide for themselves what is their best course moving forward. Do they become lab rats and they just want to do experiments and they get excited about data and discovery, or are they leaning more towards patient care and they go springboard from my lab off to medical school? And so I've had folks that do both. Probably given I've always been in a neurology department, we are very focused on taking insights from the clinic and imagining what cells can teach us about disease. And so probably the large majority of the undergraduates that come through ultimately choose to go to medical school. But sometimes we find those that we're training, folks that are, basically doing what I did when I started. I worked in 2 different labs as an undergraduate. I tried to figure out where I wanted to go for graduate school through my college experience. And so it's exciting to play that role, for these students as well and help them sort out what their next favorite step is going to be.
I know on your lab website, you talk about kind of the magnitude of Parkinson's and how many people are inflicted with this. Can you talk a little bit about that? And is it a global disease? Do we, or is it primarily maybe just here in the United States and Europe, or do we not know?
So there's evidence through the historic literature globally that Parkinson's is with us for as far back as we can find medical writing, and so Parkinson's is both global and it's been with us, for thousands of years, and so or likely has been, and so it's a global disease that affects. We used to characteristically say maybe between 1 or 2% of the population over age 65, but newer data suggests that we've underestimated that by half. And so it might be twice as common as that. And so, it's a substantial portion of the population. If you talk to folks, this is what we have, with the undergraduates reaching out to us. They have invariably a family member, whether it's a grandmother, grandfather, or someone else in their family. They've been touched by the disease. They're familiar with it. I think folks like Michael J. Fox and others have done a wonderful job of bringing greater attention to the disease. So people now, I think, have a much better way of processing what they see. In the public space, do they identify, Parkinson's disease along with, some personal history or some personal exposure to it. So, I think that the understanding of the disease in the public domain has really increased over the last 10 to 15 years. And then we do work that, as you mentioned, interfaces with Lewy body dementia and Alzheimer's disease, which is extraordinarily prevalent in the aging population. And so the questions that we ask in the lab, the things that we do that might be primarily centered around Parkinson's disease are both trying to leverage insights that we've gained from Alzheimer's disease or ALS or other disorders. We're always looking for clues that we can gain from any of these adult onset neurodegenerative diseases. And then likewise, the findings that we make and the things that we identify in the Parkinson space, we hope are not only applicable to that disorder, but that give a kind of greater insight into really these family of age-related neurodegenerative diseases.
Do we know what causes Parkinson's? And is Parkinson's a term that has kind of a portfolio of manifestations, I guess, across a group of people? Do we know?
So it's a great two-part question, right? So I'll answer the second one first. You called it a portfolio. That's a great additional analogy. I think medically, folks will frequently use the word syndrome, that there is a series of syndromes that fall under this large bucket of Parkinson's disease. It might not come in one flavor. There are debates about how should we break Parkinson's disease into different manifestations. And so we're not fully there in terms of having concrete boundaries and walls for how we would define, these different types of Parkinson's disease. But we certainly recognize that it's very heterogeneous. There's a lot of differences from patient to patient, and there may be aspects of the disease, certain symptomology or rates of progression in the disease that they kind of segregate different groups from one another, but we're still globally calling this Parkinson's disease, and we're still globally seeking to treat it the same way and imagining future therapies around that umbrella. But it may be that, 10 years from now, with greater insight into analysis of symptoms, whether it's imaging, so non-invasive modalities where we can understand more of what's going on in the brain in a patient, we may have more information where we can formally segregate into different subtypes. We're not quite there yet, but many people believe that's where we're going. So in terms of that portfolio question, you're absolutely right, asking the right question. It's not all the same. It's very heterogeneous. And we think about this a lot because it affects how we envision treatment. It affects how we think about designing and initiating clinical trials. If you want a clinical trial to succeed, whether the drug itself succeeds or not, for the trial to give you a concrete answer as to whether or not your drug or your intervention is efficacious, you're going to have the best chance of success if the patient set is relatively homogeneous. And the more homogeneous that patient set is, the smaller it can be. The easier it is to run the trial, the less costly, the less risky. We want companies to be attracted to take shots on goal to treat Parkinson's disease. And so de-risking that for companies that have potential therapies is valuable to us. And so there's lots of ways in which our ability to subtype Parkinson's disease in the future may benefit everyone. So that was the second part. Then the first part about, do we know what causes Parkinson's disease? And, to answer that question, I would say that, roughly about 10% of Parkinson's cases are genetic in nature. So they have a single gene mutation that we know is responsible for the disease, fully penetrant, meaning if you have that mutation, you're going to get the disease. And so that still represents a minority. There are many, many genes that fall within that 10%. So we have to slice up that 10% pie down to each individual gene. For those rare cases, we have a very clear sense of the gene, the mutation, and we work very aggressively on understanding the mechanism around those rare cases. And it's the hope of most folks like me that the more we understand about those genetic cases, the determined cases, the ones where I have a gene, a mutation, I can model it, I can study it, really we do that in the hope that the information that we gain around that rare 10% that is genetic in nature is actually teaching us something about the other 90%. The goal by and large is to break down the causes of Parkinson's disease into something we can actually study reliably, make some insights, make some discoveries, imagine interventions that are going to be protective, slow disease rate, or halt it altogether. And then really the hope is that with enough understanding of these individual rare genetic cases, that those strategies and the information gained there actually applies to this larger segment, and that fundamentally changed in 1997 when I was in the middle of graduate school, in what a colleague of mine, Michael Schlosmacher, likes to refer to as the pre-genetic era of Parkinson's disease. When there were no genes, we actively debated whether there was any genetic cause of Parkinson's or inheritability. What that meant was we really were just taking shots in the dark in the lab when we're trying to model the disease. We would do something and we would basically say to ourselves, I think this is what Parkinson's disease is like. And genetics fundamentally transformed the field, as it has many fields. where we started to now actually be focused on things that in rare cases absolutely caused Parkinson's disease. And that was really exciting because, you really understood where you were at that transition point in the pre and post genetic era. As soon as you had a cause of disease you could hang your hat on, people pursue that very aggressively and for good reason, right? There was a solid foundation, a good scientific premise, and what we've gained from the genetics of Parkinson's disease since then has been transformative.
You're speaking about the just kind of the decoding of the DNA that happened in the 90s and the early 2000s. Is that correct?
Well, so in 1997, the first genetic mutation that causes Parkinson's disease was published. And so there's this protein alpha-synuclein that we talk about. It is synonymous with Parkinson's disease. You hear about it all the time. But until 1997, no one was working on it. And probably in 1998, half of the labs in Parkinson's were converts now and working on synuclein. Because we quickly understood how central it was to not only the rare genetic families that had inherited Parkinson's disease, but we were finding that synuclein was becoming aggregated, misfolded, and accumulating in the brains of all of the patients. So synuclein was this wonderful example of you found a very small family that had mutations in a gene, and you dig in a little bit and you find out that the problem with that protein that's encoded by that gene, a problem with that protein existed in all of the patients that had no mutation. So this kind of paved the way to say a rare mutation can teach you something about every patient I have in my clinic. And that what that meant for the scientists like me was this is absolutely something we have to focus on. We can't look beyond this. This is central to the disease, very likely. And we were off to the races in terms of molecular and cell biology. And in the space of Parkinson's disease really was launched around that time. I was in the middle of graduate school when this discovery came out. I was not working on alpha-synuclein, as where no one, pre-publication of this paper. And I watched firsthand, asking the questions, how relevant is what I'm doing right now? This thing just came out. It's the biggest finding in the field. And I'm not working on it, and if I really want to understand Parkinson's disease, if I really want to imagine how that understanding leads to therapy, I have to, pick up and move my research into, this new area. And we find ourselves doing this, constantly. A new gene, a new major finding comes up, and if it's salient to the patient condition, if it's likely contributing to disease mechanism, you have to constantly fold that into your body of work and the program in your lab to have that more contemporary understanding of the disease. So we're constantly watching new genetic findings and new neuropathologic findings, from postmortem analysis, clinical findings on subtyping, as we talked before. How do I watch all of these things evolve and then incorporate that into what we're modeling in a lab about the disease?
So back to the portfolio and the visible manifestation of the disease. Is there any difference between people that have it genetically or those that, the other 90%? Is there kind of common across how the disease manifests, or is there some difference, or do you not know yet?
So for each one of the different genetic forms of the disease, they will be very similar. So there's one particular gene, Parkin, and so that is associated with Parkinson's disease in what we call autosomal recessive inheritance. And so that's a patient will have inherited two mutated alleles. And so they do not have a normal Parkin gene. Both copies of their gene are affected. Those patients will have very early onset Parkinson's disease. There are cases of juvenile onset, and some, but not all of those patients actually will not have the classic Lewy bodies that we think of as typical of Parkinson's disease. So those are a bit of an outlier because of the early onset. Onset can be typically in the 30s. DJ1, PINK1, two other autosomal recessive forms of Parkinson's disease are early onset. And then you have something like LRRK2 that we spend a lot of time working on in my group. The LRRK2 mutations are inherited in autosomal dominant fashion, meaning you only need to inherit one mutated copy. So only one parent has to be a carrier affected. And LRRK2 patients have age of onset that's actually typical for the idiopathic disease. So if that 90% that have no genetic basis of the disease. And so LRRK2 is interesting. It gives us something else that we're focused on because that form of the disease models the age-dependent and kind of like later onset of the disease. And so we look at each case by case. And so there's no single genetic form of Parkinson's disease that I can think of that kind of gives us everything in a nutshell and looks exactly like that sporadic patient population. But we can take bits and pieces and lessons from each one. And I think that when we work in the lab, we think about what each one of those genes can teach us about the patient population that it represents, what aspect of the sporadic disease are we going to learn in this case, and then we'll take a different lesson from a different gene. And at the end of the day, the hope is that we're, collectively as a group, we're able to assemble that into a broader understanding of all the different ways in which someone might wind up with Parkinson's disease. Because it doesn't look to me, at least, that there's one single pathway. I like to say, when you think about the all roads lead to Rome, there are many ways to get to Parkinson's disease. They're all very different in my mind. We are not stumbling upon a canonical, simple, linear pathway of A to B to C to D in Parkinson's disease. And so we have to maintain in our minds all the time the kind of different ways in which we've learned that the part of the brain affected in PD can be affected, and the different pathologies and clinical presentations of the disease. And we have to maintain all of this at the same time because of the heterogeneity that we talked about, right, in terms of that portfolio, and the genetic diversity in terms of how we can even have something as straightforward as a monogenic inherited form of the disease. Many ways to get there. And I really think that is what creates such a challenge in this disease, is that it is not simple. The more we learn, the more complicated it gets in terms of what is Parkinson's disease, how do we define it, and how are patients ultimately vulnerable to having it.
We have other scholars that are looking at Parkinson's and its relationship to the immune system, or to the, to REM sleep, or, to different kind of aspects. And what I hear you saying is it's not just Parkinson's probably just doesn't reside just in the brain. There's other, aspects of our body that is possibly affecting or a part of that evolution of the disease.
It's certainly possible. I mean, even dating back to James Parkinson's essay on the shaking palsy in 1817, he talked about constipation. He called it torpid bowels, and sleep disturbances and other things that we think of outside of the realm of a classic motor symptomology of Parkinson's disease. And so certainly there's a spectrum of symptoms that patients can suffer from beyond what we think of as just kind of this slowness of movement. And also, we've known for quite a long time that the nervous system outside of the brain and spinal cord, peripheral nervous system, is affected, denervation of the heart. There's certainly evidence for lots of things that are happening outside of, the substantia nigra, which is classically this small region of the brain that people focus on with respect to motor symptoms for Parkinson's disease. But yes, it is a syndrome, as we said, that likely affects multiple brain nuclei. We know that, and likely is affecting the nervous system and other organs in the body, outside of the brain. And so again, there's this broad effect on the brain, throughout the neuraxis, as we would say.
So we mentioned Lewy body dementia. That's not a common kind of term that people would be familiar with. What is Lewy body dementia, and how is it related, or how is it different than Parkinson's?
Yeah, it's interesting as I've read up on this. I'm not a clinician, so I don't see patients. I benefit from speaking to those that do. One that's been very influential to me since coming here to the University of Florida is Melissa Armstrong, who runs our Mangarian Lewy body dementia clinic here. And, Parkinson's patients initially will be sent to a neurologist, often for movement problems, falling, tripping on carpets. These are the things that might be first noticed. But ultimately, there are psychiatric symptoms that develop later on, and as well as can be dementia. And so what clinicians have decided is that and this relates to your question about the portfolios under which Parkinson is, that if a patient begins with symptoms of dementia before there's these formal motor problems, or if substantial cognitive decline in a particular form of dementia arises within 1 year of motor onset, they will actually be broken out and be called Lewy body dementia. And so we're taking these patients that have features of Parkinsonism, but if the symptomology for that particular patient begins cognitively as the major feature before movement, then we're going to call them something else. And actually, it turns out that they respond better to different medications, and there's other things that they might be working with clinicians on in that kind of subcategory, that subportfolio you talked about. So what clinicians are finding is the timing of the different kinds of symptoms that a Parkinsonian person might experience may be useful in this subclassification. And so when we look at these patients that are pulled out because they either have dementia first or dementia within 1 year of motor symptoms, right? So this is a dementia first subtype. When those patients pass and go on to be analyzed at autopsy, and they do a neuropathologic examination, we find that they have Lewy bodies, and so they have this classic neuropathologic feature of alpha-synuclein inclusions in the brain. But they very frequently have, in addition to that kind of Parkinsonian pathology, they also have pathology very typical of Alzheimer's disease. And so when we think about Lewy body dementia, what makes it special, what segregates it, there's the clinical aspects I mentioned, which is a dementia first, but frequently superimposed with motor throughout most of the disease course. But they also have what is going on in the brain is actually the deposition of multiple proteins. There's a pathology that looks like, we could colloquially say, Parkinson's and Alzheimer's at the same time. The dementia can be distinguished by a clinician from the kind of memory deficits in classic Alzheimer's disease. So they have tools behaviorally to distinguish those forms of dementia, but it really is about timing. And then at the end stage of the disease, what we're finding is actually what's going on in the brain is very different, where they have this superimposed pathology of Parkinson's and Alzheimer's. And that was one of the questions that I really started to get excited about and interested in, because my own training, I began in graduate school doing Parkinson's disease, and I sought training in cell biology and molecular biology in Alzheimer's disease as a postdoc, because Alzheimer's was about 10 years ahead in terms of the technology, the identification of genes, and the application of those, mouse modeling, everything. And so Lewy body dementia as this subtype became a very attractive means for me to think about, how do I leverage both of those training experiences and really everything that I had learned in both of my training environments when I was younger, and coming here and talking to Melissa and about the identification of, interestingly, two genes, the two major risk factors for Lewy body dementia. One is primarily thought of as a risk factor for Parkinson's disease, and one was primarily thought of as a genetic risk factor for AD. And you take these two particular risk factors, and when you look at that subtype of Lewy body dementia, and those two genes are playing a major role in risk of Lewy body dementia. And that's what I got really excited about, is what is this, the Venn diagram from PD and AD, and that ApoE gene and GBA1 gene are at the interface of these two, where Lewy body dementia sits as a disease where patients are basically suffering from both pathologies, the Parkinson's and the Alzheimer's. And what are the mechanisms that underlie that? Why is it that GBA patients, for example, that for many years we thought of as a very, very strong risk factor for Parkinson's disease, why do they suffer from a greater risk of dementia with their Parkinson's? Why is it different from a LRRK2 patient that is genetically determined to get Parkinson's disease? Their dementia risk is not equal. And so the pathway from GBA to Parkinsonism or Lewy body dementia involves a greater risk for dementia. And at the cellular level, at the mechanistic level, that's the kind of thing that I get very interested in. These subtle differences mean a lot to the patient. You know, what is the mutation that you have and what is the likely disease outcome? And if we can understand that, we start to understand the subtypes of each disease in parallel.
You haven't, and maybe you have, but this is something new in the last few years that you started to concentrate on. Is that correct? And then how many other researchers, is the group pretty small? Or are you kind of unique, or are there kind of a growing amount of researchers that are looking at this intersection?
I think there's a growing interest there. It really began with folks that were, pure Parkinson's clinicians that were studying their GBA carrier patients that recognized initially higher rates of dementia in that cohort. So Clement Schertzer, a former colleague of mine at Brigham and Women's Hospital, had done some of this early on. And it takes time, right? So the clinicians see the patients. As time goes on, brains come to autopsy. We can study the brains more deeply of the patients themselves. We start to come up with some clues, and we put together the clinical picture. We put together the neuropathologic analysis. And then we can start to look in our cells and our mice, and we can ask mechanistic questions. But for something like this, we're really waiting for these subtle insights and the subtle differences that the clinicians and the neuropathologists recognize. And then we go further. And so I think it kind of begins with, for at least my group, it really begins with, what are the questions that a clinician would have but can't ask? What do they want to know, but they can't do the experiment in a patient? What are those mechanistic questions that they're fascinated by? Can we be challenged to come up with an experiment to add some level of information to that? And so, as a major risk factor for Parkinson's disease, GBA is very interesting. I think it's very tractable in the lab to study. And so I think there's benefits there, that there are certain things that GBA models do that are consistent with the human disease and therefore warrant investigation. And so I'm certainly not alone in my interest in that. I might be more unique in having that Parkinson's and Alzheimer's kind of dual training background. But I think a lot of folks have a similar interest, and they may just approach it from a different angle, than my lab would.
So what are the greatest challenges that you're kind of faced with then as you do this research and search for these answers to the questions that you have? Is it funding? Is it technology? Is it access to a variety of tissue samples? What are kind of the big challenges that you have? And is there help that could help you accelerate some of this research?
So, if you ask any scientist, they're going to say funding is always a challenge, and it is, but that's universal across, whether you're a cancer biologist or, whether you're someone like myself. I think in our particular field, one of the greatest challenges we have is how well our animal models that we use that are so critical to asking systems type questions, how well do those animal models faithfully reproduce the human disease? And in the context of Parkinson's disease, in my opinion, they're a bit limiting. And that's one of the limitations that we have. These are age-dependent neurodegenerative diseases. Even folks that have mutations that are genetically determined to get Parkinson's disease will live symptom-free for many decades before there's any problem. And understanding the aging component, what is that doing and what role does aging play in the disease process is really important. And so that is the importance of in vivo studies, and our reliance on animal models is largely around wanting to understand that part of it, wanting to understand how aging is contributing. In addition, we can do lots of things in addition, and my lab uses lots of stem cell models, and we can make neurons, brain cells, and glia and immune cells, and we can study them in a dish, but there's really no replacement for, a multi-year-old animal where all of the cells are in tissue, talking and touching one another as they would in the human brain. So that's a wonderful goal to achieve and to have that, but the models really don't replicate Parkinson's disease the way we'd like. And so that's a major limitation. And frankly, it's one of the reasons that my lab has gotten more and more reductionist, is that we're focused on answering questions that we can and recognizing limitations in front of us. And so I think one of them is the animal models. I'm fortunate. We have right down the hall, I've got Stefan Prokop, who runs the UF Neuromedicine Brain Bank. And so, our group and groups around us have good access to human patient tissue and mouse brain tissue to evaluate models, to test our theories. If there's a certain thing we think is occurring in a Parkinson's patient brain, if we have a novel discovery in the lab, then we can go to the brain bank and we can start to ask whether any of the things that we're seeing in our cells or our mice are happening in actual human disease. That's the benchmark, right? And so I think that brain banks are very important nationally. Access to tissue. There are many around, and what you find is that people will kind of congregate around access to that tissue, because you always have to go back, and ask, how grounded is my hypothesis to that actual human disease? You don't want to go, off the reservation and be studying some kind of red herring that only exists in your dish of cells. We want to be focused on the human disease. And so brain banks really are valuable, and they give us that kind of way to calibrate back to human disease. Conversations with clinicians, again, I'm a PhD scientist, but I've spent my entire career in neurology departments. And I find that fun because I don't see patients, and how can I claim to study Parkinson's disease if I don't see the patients and I don't interact with the clinicians and have that kind of grounding on the clinical side of it? So there's lots of things about the medical school environment, I think, that is amenable to kind of recalibrating back to the disease.
You mentioned it's an age-related disease, and it's probably just because of the age that I find myself at, which is aging. But is it aging, or is it the behavior that we kind of adopt as we age? And I'm very fascinated with these super agers and some of the research that's going on. And I wonder, my wonderment is there just kind of a mindset and behavior that, of course we age, but that may accelerate that aging? Is it or not, right? Are we doing things in our 40s and 50s that if we didn't do them and if we kept pushing like we were 20 and 30, would it really be an age-related disease, if that kind of makes sense?
Sure. It's an interesting question. I think that on the cognitive side of it, there's a lot of data to suggest, and people espouse the model of use it or lose it. And that, later in life that maintaining mental challenges and dealing with complexities, social interactions. I mean, we know a lot, of this from human behavior and animal studies, that the more impoverished your environment gets. So if we extrapolate to your social network and the numbers of different types of people that you interact with and your social engagement, as that withdraws as we get older and the kind of our worlds get smaller, that certainly could compound age-related changes in the brain and affect cognition. And so I think that in the context of Alzheimer's disease, there's a lot of folks that espouse, kind of like, mental games and activities to stay stimulated. I would argue that I think the social component of that is probably just as important in terms of kind of maintaining, mental focus. you have to remember the brain is what we call post-mitotic, right? We by and large don't make new brain cells, over time. And so you've got, and much of the wiring in the brain is probably complete maybe around age 25. And so you've got your complement of synapses and your full complement of brain cells at that age. And then what happens as you age and how effective are the cells that remain? Definitely an aging component. And I think we all want to know, what can we do in that context, which we can't stop? What are we doing behavior-wise that could offset age-related changes or decline? So I'm not sure about the motor. It's certainly interesting to think about. I'm sure we are much more sedentary as we age. I'm sure that all kinds of things in terms of muscular load and what we're doing, not just the number of steps you take, but the actual intensity of physical work and moving, is dramatically changing in those later decades. And does that have some impact on your motor system as well? We believe that the cells in the substantia nigra, these that are most vulnerable in Parkinson's disease, likely are dying at a rate about 1% a year into adulthood. And so there's some very slow decline. Parkinson's patients have a dramatic steep drop off or accelerated decline in those cells. But that's not to say that there aren't ways to compensate for that, even in terms of motor. And so I don't, certain people look at dance and exercise for Parkinson's patients, but I think it's unclear whether is that simply allowing the brain to compensate for a loss and improve motor outcome and coordination, or are we actually protecting the remaining cells we have? I think that's still an open question.
Yeah, more to come on the study of these superagers, because it's fascinating. Not only their brains, but they're picking up skills and they're running marathons at 98, and it's pretty amazing, kind of physically and mentally, what they're doing. And it doesn't mean that they're super healthy either. It's just that they're kind of redefining aging, which is fascinating. What about technology? You said your lab's become kind of reductionist and really answering the questions. Are there technologies that are emerging? I mean, you hear through the news debates around artificial intelligence or machine learning, robotics. Are there technologies out there that that you have started to adopt, or considering adopt, to help you with this pursuit?
Yeah, so I mean, we are by no means, the pioneers of these technologies, but rather we're borrowing them. But my lab does a lot of stem cell biology, where we are using pluripotent stem cells and differentiating them into different cell types of our interest by our design. And so that has fundamentally transformed the field, where I can say, I need a dish of this kind of cell type from this patient, and the ability to do that. And then in addition to the stem cell revolution is also gene editing. The fact that I am no longer reliant, if I want to study a particular gene mutation, I'm not reliant on getting a skin biopsy from that patient and then reprogramming their skin to stem cells and then their stem cells into brain cells or glia or what have you. Gene editing allows me to read a paper where someone has found a new mutation in some very obscure patient population. Gene editing allows us to introduce that mutation into an otherwise healthy cell. And I don't have to go across the globe to find that actual patient. So I can recreate these mutations at will, and I can recreate them in stem cells, which then means at will I can have a novel cell type with the novel cell mutation that someone else has identified as causal for disease. And so that expedites everything, is much faster now. And also it increases rigor, because many people can now do that. So rather than one person having cells from an affected individual, now everyone can do the same experiment. And we can ask, are we all seeing the same thing? Can we replicate each other's data? So now we have rigor. Now we have greater confidence in the findings, because if it's real and I see it here, then someone in another country can do the same gene editing, and do the same experiment, and they should see the same thing I do. And so we're not dependent on rare sources of tissue, cells, patient information. We can do this all de novo. So that's been really huge. As far as machine learning and AI, which is a big deal and people talk about all the time, there certainly is the opportunity if we blend machine learning with, for example, microscopy, for watching a live cell, react to an environment. There are likely things that are happening in that cell that the human eye doesn't perceive and cannot track and interpret that, every time I look at a patient cell, it does X. And every time I look at a healthy cell, it does Y, and kind of dissect these things in a video-based system where machine learning can take these images and information from something even as reductionist as staring at a single cell. And it gives us the opportunity potentially to see things that would be below our perception level. And so I'm hopeful that in that context, that machine learning can help us to add to the list of phenotypes, the things that we attribute to Parkinson's-like behavior of a cell. I'm hopeful that machine learning and AI adds to that list things that would take another 20 years for someone to anecdotally observe, staring at the microscope. So I think there's lots of opportunity to dig deeper into data, particularly image data, in my mind, to find new things to hunt after related to disease.
Yeah, that's great. Can you just spend a couple moments on stem cells? Because a few years ago there was a lot of debate, a lot of concern, a lot of misinformation. Can you talk about there's different kinds of stem cells and what the type of stem cells you are using?
Yeah. So, it's interesting. And it used to actually be a question I would get on airplanes whenever I traveled, and it came up, that it would be a very, politically charged conversation around stem cells. And so, once upon a time, we were limited to stem cells that had originated from their fetal stem cells. And so there was a concern around basically generating a marketplace for, abortion as a source of fetal stem cells. And what would this mean? Because the potential for the research was clear, and how we dealt with the demand that everyone would want to work on stem cells was unclear. We didn't know how to deal with that. And so here, in the States, there were certain stem cell lines that were approved for use at NIH, and then the creation of others was banned from NIH funding. There were limitations placed on what we do. And then a researcher, Yamanaka, had discovered, to certainly my surprise, that you only needed 4 genes to be expressed in a cell type. We call them now the Yamanaka factors. 4 genes to reprogram a cell to revert back to a pluripotent state, to make it become a stem cell. And this was a massive transformation in the field, because what this meant was I no longer needed fetal stem cells from an individual to have a representation of stem cells from that patient. You could take a skin biopsy. You could take a blood draw. And now it's expensive, but anyone can take blood or skin, and they can send it off to a commercial facility which will reprogram those cells and give you back pluripotent stem cells. These are what we call induced pluripotent stem cells, because they did not begin life as a stem cell. They began life as a red blood cell or a macrophage or a skin fibroblast. But we reprogram them into this induced pluripotent stem cell. And from that origin, I can now make a brain cell, a glia, an immune cell, a skin cell, a bone cell. And so, having to work around the limitations of fetal tissue has led to now what is a universally available technology, which is iPSCs. And then if you couple that, as I mentioned earlier, with then genome editing, now I don't even need the blood of a specific patient. I just need to know what the mutation is, and I can do it in my own lab. And so this has really brought this technology virtually into anyone's hand. So I think that the combination of the Yamanaka factors, a Nobel Prize winning, discovery, and genome editing now has really made it so that we don't even talk about the past limitations, because they never, that technology of fetal stem cells would never have brought to the wider scientific community access at the scale we have now, where I can buy from Thermo or a variety of sources induced pluripotent stem cells. And so I always make it clear to folks when I talk to them now that what we're talking about are cells that come from a skin punch, by and large, or a blood draw. And I think that makes people really excited that these are the kinds of sources that are easily accessible and are not tied to the baggage of the past that we had before.
Yeah, I really appreciate you going through there, because, things they get out there, they get, in the media, they spread, and people don't typically check back on where science has progressed and how far it's progressed. And I think there's still a lot of misinformation out there around stem cells and what they mean and where they come from and the potential. I'm going to put you on the spot just a little bit here. You talked about some of the discovery. One of the Karen Toffler Charitable Trust is very very proud of some of the work that you have done in the last couple of years. And what can you describe, like, what you use the money for, and where it kind of got you today? You don't have to spend a lot of time, because this is not a plug for the trust, but I do know that you're pretty excited about some of the work that you're doing and where it's going.
Sure. So I guess off the top of my head, there are two things that come to mind that we've done since coming here and being associated with the Toffler Trust. One was a colleague of mine, Ben Giason, studies a lot about how the protein alpha-synuclein accumulates and aggregates in Parkinson's disease and Lewy body dementia, and is very good at making unique antibodies and reagents to try to identify in that human tissue, the bona fide disease, what is going on, right? So Ben is a great example where he makes observations in the human disease, and then that raises a whole bunch of questions, how do we get there? Chicken or the egg type of questions. And one of the observations that he had made is that there are particular truncations, so places where alpha-synuclein is cut into smaller fragments. And he has identified different smaller fragments and which ones show up in Parkinson's disease, Lewy body dementia, other disorders. MSA is another synucleinopathy. And, when we look at the end stage of the disease and we look at a brain, after autopsy, we can ask ourselves, did it start as a small truncation of synuclein, or was it there and it just got eaten away at and nibbled by proteases that chop things up after it formed a Lewy body? So Ben is looking at these in Lewy bodies. And we don't know whether, it started as a full-length protein and it got nibbled on, or was it cut first? And is that cut actually contributing to the disease? Is this chopping up of alpha-synuclein important to making disease progress and get worse? And so I thought it was really interesting. He had, particular truncations that he saw that were very common in Lewy body dementia. So my lab started looking into GBA mutant cells that we had used CRISPR genome editing to create. So we created our own little model of GBA, which is a risk factor for Lewy body dementia. We started to study, how do those cells process alpha-synuclein? And again, these cells, they don't live 60, 70, 80, 90 years. They're not forming Lewy bodies. There's nothing age-dependent about them. This is all very, very, very early stage, imagining what's going on in the brain of a patient decades before diagnosis. And what we found was that GBA mutant cells would produce way more of these truncated synuclein species than wild-type cells. And so this tells us that it's very likely that these truncations that Ben had identified in human tissue are occurring very early in the disease process. And so we're starting to try to understand when in the process we start to place certain pathologic events. And so synuclein truncation, I now believe, is earlier than I used to think it was. And so using GBA as a model of Lewy body dementia, partnering with him, we started to understand. Now we're looking at how are the truncations made. And so that's kind of the next piece of the story. How are they cut? Why are they cut? Who is cutting them? And so we're working together on that. I have a graduate student, Liz Chapman, doing that. And so that was really exciting because it was inspired by, the human brain data that Ben had. And then the other thing, again, related to GBA, we've really focused our work that was supported by the Toffler Trust on GBA as a model of risk for Lewy body dementia, is that GBA as a protein traffics to the lysosome and is linked to Gaucher's disease, which is a lysosomal storage disorder. And so classically, most of the effort that focuses on the GBA protein stares at lysosomes in the cell. And we've been very surprised to see over the last few years that actually the GBA protein or cells that are deficient in the GBA protein have dramatic effects outside of the lysosome. And so we're looking at ER and Golgi and other organelles in the cell that have really been overlooked in the past. And we believe that these upstream organelles in the cell are actually contributing to a lot of the pathology that we see in GBA cell or animal models. And so we're working with now in the grant that we talked about is really pulling together a team of 4 different PIs, including myself, at the University of Florida to tackle this question of how does GBA mutation not just contribute to a synucleinopathy and something that looks like Parkinson's disease, but really more broadly, why does GBA look different than the other PD genes? Why is it contributing this extra risk of dementia? What other element is going on in a cell or in a whole animal that is adding that risk of dementia that we otherwise, don't expect to see in a pure Parkinson's gene? And so we think that looking outside the lysosome in the cell is starting to give us a broader picture.
Yeah, that's very exciting because if you can get those, that understanding, at some point there might be diagnostics and possibly therapeutics down the road for that. When you look out the next 5 to 10 years in your research and what you're doing and what the community is doing, what are you the most excited about?
I think that, one of the things, and I hope to take better advantage of this myself, we haven't, I was just talking about this with one of my fellows earlier today. The stem cell technology that's available allows us to ask about what we call selective vulnerability. So I can take a particular stem cell of a particular type, and I can transform that into or force it to differentiate into a cell that is relatively unaffected in Parkinson's disease, or I could force that cell to differentiate into something like a dopamine neuron of the substantia nigra that is dramatically affected in disease. And as I said earlier, the mouse models have really failed to yield us that kind of information. This selective vulnerability is not replicated sufficiently in the mice. And so I really hope that we need to get better at this technology. The field is going to evolve to make the technology more accessible. And I think the selective vulnerability, why is the substantia nigra so vulnerable in Parkinson's disease? That's going to be critical. It's the same kind of question we would ask for ALS. Why are motor neurons so affected in ALS? And I think that the evolution of stem cell biology and simplifying techniques and protocols is going to put that technology to ask those fundamental questions. Why this cell in this disease? The more people that can do that kind of work, the faster we understand the vulnerability, and then the better models we have to test therapeutics to stop those cells from experiencing that stress that's leading to that vulnerability. And so that's one of the things that I'm most interested in seeing come about is better access and better models towards that. I don't think we're going to see that in mice per se, but I think there's tremendous potential to make this scalable to more investigators using stem cells.
The future is always unknown, but do you think in the next 10, 15 years that this Parkinson's, it won't be cured, but will there be something out there that will, significantly help those that are inflicted, or just kind of stop the disease or slow down the disease? I mean, what is that time horizon, just to give us some idea?
So there are numerous clinical trials ongoing that are targeting pathways linked to genetic forms of the disease. Those trials will be complete. We will have an information, and I think 5 years from now, we'll be able to sit back and say, in a therapy targeted for a small population of inherited Parkinson's disease, how did we do? Did we slow the disease? Did we stop it? Where are we when we've got a very specific pathway that we know is causing the disease? Are we able to interfere with that disease? That will be easily within 5 years. I think the 10-year question is, of all of those efforts, for each one of the rare genetic, forms of disease that we're trying to treat now, in 10 years, we will know whether any of those are actually going to benefit that 90% that do not have a genetic mutation. And I certainly hope that many more targets will be nominated in that first 5 years for the sporadic PD population. But certainly the efforts that are ongoing now, in 5 years, we will know how well we're doing for the small population they were designed around. And we should be very hopeful around the ones that are targeted around genetic forms of the disease. And then the 5 years thereafter, we should have solid information on how many of those are broadly applicable to most patients, to targeting that majority.
That's wonderful. Well, I hope that's the case. And Al Toffler would say that, the future always comes at us faster. So maybe it will come at us faster than 10 years. And that would be wonderful. Well, Matt, as we kind of wrap up here, where can people get a hold of you, get in contact with you if they have questions, want to collaborate? How would they get in contact with you?
Sure. Well, they should be able to find me on the University of Florida website. Just look up Matt Lavoie, UF. My email address is mlavoie@ufl.edu. You had mentioned LinkedIn. There's other places. And so I shouldn't be too hard to find. And I welcome comments or collaborations or ideas following our conversation today.
That's great. Well, I really appreciate your time. I always, I appreciate the update on what you're doing. I'm very fascinated with this kind of cross look between Parkinson's and Lewy body dementia. And I think, those connections there, and that just really kind of precision of trying to understand some of these inflictions are just really, really important. So thank you very much for the dedication and what you do and who you're mentoring in your lab and the people that you're in contact with. So thank you very much.
Oh, thank you for having me.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.